In lab exercise 9, we are going to learn how we quantify evolution. In other words, we're going to use mathematics to demonstrate how a population is changing over time. So if you remember from last week, we defined evolution as a change in a population over time. Now in our lab last week, we looked at the change in the frequency of a trait. We were looking at how fur color changed in response to a selection pressure in an environment, or how the frequency of fur color changed in response to genetic drift. In this week's lab, we are going to look at all of the mechanisms of evolution, the 4 mechanisms that cause evolutionary change. And we are going to calculate allele frequencies and genotype frequencies in order to determine whether or not our population is actually changing over time. So this gives us a mathematical way to demonstrate that change has occurred. Or alternatively, maybe change has not occurred, and we can demonstrate that as well. So the first thing we need to learn how to do is to calculate allele frequencies. So there are 2 variables here. The p variable, the lowercase p, is going to stand for the dominant allele. So let's say that is allele capital A. And it can be any capital, it doesn't have to be A. We'll just use that as an example. So the frequency of the dominant allele is going to be equal to the number of dominant alleles divided by the total number of alleles. Now keep in mind that each individual has 2 alleles. So let's say we have 50 individuals in our population. That means we have 100 total alleles. So another way to note the frequency of the p allele is to say p divided by p plus q. But if you remember that we're looking at total of alleles on the total number of alleles on the bottom, you'll be good. So the problem will either give you a number of dominant or recessive alleles directly, or you might have to calculate the number of recessive alleles. And we'll talk about that part in just a minute. The lowercase q is going to represent the frequency of the recessive allele, lowercase a. In this case. Again, it can be any lowercase letter. So the frequency of Q is going to be the number of recessive alleles divided by total number of alleles. In other words, Q divided by P plus Q. Now I just noticed that I made a little typo up here. I meant to say P equals the frequency of the dominant allele. So keep in mind we're dealing with frequencies. Now a frequency is always less than 1. And also when we're dealing with frequencies, the total for all of our frequencies is going to equal 1. So P plus Q is equal to 1. So this is important because if we find P, then we can calculate Q without going back and redoing our calculation. So keep in mind that P plus Q is always equal to 1. Now this leads us to something called the Hardy-Weinberg Equation. So we can do something called a binomial expansion, and you may have done this in algebra. You don't have to know how to do it, but essentially what we're doing is we're squaring both sides of this equation. And when we square both sides of the equation, we're basically saying the frequency or the chance of getting one allele times the chance of getting the other allele. So we end up, after we square this, getting p squared plus 2pq plus q squared equals 1. This is our Hardy-Weinberg equation. This is the important equation for this unit, along with this equation here. So these equations are directly related, and you have to know how to use each one of these in order to calculate these problems. So the one thing I tell my students is, as soon as we start Hardy-Weinberg, you need to memorize what these variables mean and how to find them. P is the frequency of the dominant allele. You get that by dividing dominant alleles by total number of alleles. Q is the frequency of the recessive allele. You get that by dividing number of recessive alleles divided by total of alleles. You have to memorize that. If you don't, you will be confused the whole time. All right, so let's talk about this Hardy-Weinberg equation. You should have already watched the lecture video on this, so this should be a review. But P squared is going to be the frequency of homozygous dominant genotype. In other words, the number of individuals in a population that have the homozygous dominant genotype, or AA. 2PQ is the frequency of heterozygous genotypes, or big A, little a. Q squared is the frequency of homozygous recessive genotypes, or AA. Now, quite frequently when we're doing problems, we're actually given this Q squared, and you can use Q squared to determine Q. And you do that by taking the square root of Q squared, and that is equal to Q. And that is one of your values that you may need to determine your allele frequencies. But we're not going to focus on that right now. For now, I need you guys to understand this equation, and I need you to understand the Hardy-Weinberg assumptions. Now, I'm not going to go over these because they are written explicitly in your lab. They're in the lecture. But just know that when all conditions or assumptions are met, no evolution is occurring. But if any one of these are violated, and most often it's natural selection, but not all the time, if any one of these are violated, then evolution is occurring. That means if any of these assumptions are violated, the allele frequency in a population is changing. So the first thing I'm going to say about this lab is to go back and reread the introduction. Work the math problems that the introduction gives you. Pay attention to the variables you're calculating and to what they mean in the population. Pay attention to where we're getting those values and what information we're using in order to calculate them. Then your lab manual suggests that you purchase beans. So you may purchase beans. I would personally prefer that you purchase beans and actually do this simulation. However, I realize that most of you are not going to do that, and so the data is provided for you in Blackboard. So go through that data carefully. As in all the other labs, it will step you through the process. So for all of these sections, you're going to have a bag of 2 different beans. Each bean stands for a particular allele. And remember that each individual has 2 alleles. So for every 2 beans you pull out, that counts as 1 individual. So your manual will explain to you which beans are which, but if you pull out 2 dominant beans, that's a dominant individual. If you pull out 2 different beans, that's a heterozygous individual. If you pull out 2 recessive beans, that's a recessive. So you'll go through and you'll pull out some number of homozygous, heterozygous individuals, and you'll need to calculate the total number of individuals. So you'll tally up the number of individuals here and write it here. Then you need to calculate the number of alleles. So in the first simulation, we actually have 25 of each allele for a total of 50 alleles, which means over here we actually have 25 individuals. Then you are going to use the information in the top table to calculate the bottom table. So remember that p squared is equal to the frequency of individuals that are homozygous recessive. So you are going to take the number of BB individuals divided by total number. For heterozygous, we're going to do the same thing for each generation. For homozygous recessive, you'll do the same thing there. Now, over here to calculate P, you're going to take the number of dominant alleles divided by the total number of alleles. Now, you will need to actually calculate this out. Don't write the math problem there, write the actual answer. I'm looking for the actual numbers. And then you're going to answer a series of questions. Now, you're going to have to look and see, did the genotype frequency change across generations? Why or why not? Keep in mind that sometimes things don't behave as you would expect due to chance. Did the allele frequency change from generation to generation? Yes or no? And then, did evolution occur? Remember that evolution is change in allele frequencies over time. Did the allele frequencies change across all 3 generations? Now, if they fluctuate between 1 and 2 and then 2 and 3, that is a change. Even if in one generation by chance they happen to not change. If they change though, evolution did occur. So the rest of this is just repeating 4 different violations of Hardy-Weinberg. The one that we are not doing here is random mating, the violation of non-random mating. And the reason is that non-random mating actually does not cause evolution to occur. It does not cause gene frequencies to change over time. The other one we're not doing here is mutation, and that's because it just gets complicated. If we introduce a mutation, we introduce a third allele, and we need a slightly bigger equation to deal with that. So we're not doing random mating, we're not doing mutation, but we are doing the other 3, natural selection, genetic drift, and gene flow. And then at the end, you have a few questions to answer. Now, I want to direct your attention very quickly to question number 28, because this is the most missed question on this lab. If you have watched the practice problem videos for Hardy-Weinberg and gone through any of the practice material or the homework, you will notice that I tell you the first step in doing Hardy-Weinberg problems is to identify if you are given alleles or phenotypes. Now, if you are given a phenotype, you're given a physical characteristic about the organism. So if you are given a phenotype, you can infer a genotype frequency. Because remember that genotypes correspond with phenotypes. So if you are given some information about a recessive phenotype, that corresponds to Q squared. If you are given information about a dominant genotype, that refers to both P squared and 2PQ. If you're given a dominant genotype, you cannot use it to calculate the frequency of P because you've got this little Q hidden in there. But if you are given a recessive phenotype